Hi, my name is Brianna Melton, and we will be presenting on Paul Ehrlich, Emil von Behring, and Robert Koch. So first, some facts about von Behring. His birth date is March 15th, 1854. He was the eldest son of the second marriage of a schoolmaster, and he was the eldest of 13 children. He passed away on March 31st, 1917, and he was born in Honsdorf, Poland. I can't say that second name. He passed away in Marburg, Germany. And in 1896, he married 20-year-old Elsie Spinola, who was the daughter of a director of a charité at Berlin. So the problem here was that diphtheria was an infection that's caused by a cyanobacteria that was spread through respiratory droplets during World War I between soldiers and military personnel. And they would come also into direct contact of that within the tools and any weapons that they were using. And then we were also looking at tetanus, which is caused by an infection, a bacterium that's in the soil. And obviously, this is a really big deal with the First World War because of the trenches that were built and where the soldiers were having to live for long amounts of time. So how was this problem fixed? As you can see here, it went into a good bit of detail, but essentially von Behring and Koch actually worked together to discover what would actually work to kill off this bacterium. They also joined with a researcher from Tokyo University on providing the substance that would show what was resistant to tetanus in rabbits. And that actually showed that pretreated mice became immune and did not show any symptoms of tetanus, while the control animals died shortly after being infected. And this was after the vaccine was given to these animals. So they showed that the antibodies actually produced by one animal could be removed and used to immunize other animals, and this is where the vaccine was actually born. After this, they also decided to conduct an experiment on guinea pigs using this therapy, and it actually showed to be effective against this bacterium that we were seeing. And the ones that were injected with this vaccine actually did not become ill. So that's where this was first born. How did this immunologists actually relate to the others? As I said earlier, Koch actually worked with von Behring on multiple occasions. They actually were able to work together at the Institute for Infectious Diseases in Berlin, and they were able to actually meet Ehrlich, and they became a team that really wanted to research and really provide an opportunity for people to have vaccinations that may have not been economically stable, and they were not able to have the best access to those vaccinations. So they really wanted to provide for those who could not actually provide for themselves. So today, here's some statistics about what we're seeing for tetanus and diphtheria. Thankfully, there's only been 2 reported cases of diphtheria in the U.S., but the disease continues to spread in third world countries. And as you can see here, in 2016, countries reported over 7,100 cases, and this came from the World Health Organization, so an extremely reliable source, but I'm sure that there are many cases that likely go unreported due to lack of healthcare access. And for tetanus, we're obviously seeing a decline because we do have a 10-year vaccine that's given as a booster shot, and we as nursing students have to get this before we are able to continue in our program. So these are very important just to keep us healthy and to keep this infection from spreading and to keep us as immune and as resistant to it. So next we're going to talk about some of his awards. In 1893, he was awarded the title of professor, and then also became the Greimer, I'm not going to say that second name. Essentially, he was the chancellor of a college. He was also he had an honorary membership in Italy, Turkey, and France. And then, of course, the biggest thing is 1901, he was awarded the Nobel Prize in Physiology. He was also raised to nobility in Germany. In 1903, he was elected to the Privy Council with the title of Excellency, and he also received honorary memberships in Hungary, Russia, as well as orders and medals for Germany, Turkey, and Romania, so he was a very well-liked immunologist. And he also became an honorary freeman of Marburg, where he passed away.
Hello, my name is Noah Elkins, and today I'll be talking about Robert Koch. He was born on December 11th, 1843, in the Upper Harz Mountains in Germany. He died on May 27th, 1910, in Baden Baden, Germany. He was born to Hermann Koch, a well known mining engineer at the time, and Matilda Julie Henriette. He was married twice. His first wife was Emma Adolphine Josephine Fratz. With her, he had his only daughter Gertrude, and they were married for 26 years. And when they got divorced, yes, less than a year later, he married his second wife, Hedwig Friedberg, and he was married to her until his death. Among Koch's many achievements, he is most well known for his postulates. If you don't know, these are just basic guidelines for researchers to follow when creating relationships between microbes and diseases. Koch said himself it's not a perfect system and it doesn't work for every microbe, but it works for the majority of them. It's as follows. The microbe must be present in all infected animals, but must not be present in healthy animals. Second, the microbe must be isolated from a diseased animal and must be able to be grown in a culture in a lab. From there, the same pure culture must be able to infect animals, most importantly, healthy animals. And the last part is that the microbes collected from said infected animals must be able to be collected and regrown in pure culture once again. Koch worked on several problems throughout his researching time. The main three were anthrax, tuberculosis, and cholera. We'll be discussing these more later in later slides, determining how he solved these, but the basic overview is for anthrax, at the time it was decimating the local farm animal population in the German town he was staying. People had an idea that a small microbe was causing the disease, but however, they could not find it, so Koch set out to find that. Shortly after that, he began his work on tuberculosis. While it was known that TB was caused by an infective agent, it was very hard for people studying it to see it because it's only a third of the size of anthrax, and it wasn't easily stained. So this was arguably Koch's greatest challenge during his researching career. Cholera, the last major event in Koch's life, was uh, basically him performing the same research as he did with anthrax and tuberculosis. In this case, Koch was sent to Egypt to research a recent outbreak of cholera. It had become an epidemic, and in his time there, he was able to find some interesting bacteria, but the epidemic ended before he could really finish his work. We'll discuss more of that later. So going back to anthrax, to study this, Koch set up a lab in his own house, and it was at this place that he performed research in order to discover the cause of it. So to do this, he set up a test where he inoculated lab mice with the blood from the spleen of infected animals and inoculated some lab mice with the blood from the spleen of healthy animals. Obviously, the mice that were inoculated with the anthrax bacilla infected blood died, while the other mice survived. Koch also grew anthrax bacilla in culture, and he discovered that the bacteria could still cause disease even though it had not been grown in an animal. This research led to the creation of Koch's postulates and also agar jelly, which we still use in lab today. Koch was very determined and very persistent, and he repeated this process over 20 generations of mice before announcing his findings later in 1876. Soon after this, he started working to find the causative agent for tuberculosis. Some at the time believed that TB was inherited through genetics. However, Koch, among some of his other peers, believed that there was also a causative agent for this as well. However, they had been unlucky in finding any successful proof for this. Koch was able to successfully grow the tubercle bacillus in animals and on slides. However, he was not able to easily see the bacteria on the slide, as the bacteria was quite small and would not accept any conventional dyes. Koch eventually was able to properly stain the bacteria and was able to see blue rods. And when he looked in the animals that he infected with the disease, he found the same blue rods and the same yellow tubercles growing out of them. So at this point, he had not only created agar jelly, he had also pioneered staining techniques, which we still use today in lab. From there, Koch made his way to Egypt. He had heard about a major outbreak of cholera occurring in the Nile Delta. During his time there, he had found a potential cause, but before he could perform any other tests, the epidemic slowly died down and eventually ended. After he returned to his hometown of Berlin, he quickly determined that he had made the right choice about the bacteria and that the bacteria he had collected was the cause of cholera. To prove this, he traveled to India. At this point, the disease had been endemic for years and had really affected the people there. It only took 2 months for Koch to discover that the bacteria was found in 70 cholera patients that he had studied. The bacteria was not found in any healthy patients he observed, still following his postulates he created. Koch also determined that the cholera was being caused by the dirty water that was then drank by the population, primarily public water sources in the city and dirty lakes in villages. Some major awards that he obtained during his life: he was elected as a foreign member of the Royal Society in 1897. His biggest award came when he won the Nobel Prize in Physiology and Medicine in 1905. He won the Order pour le Mérite in 1906, and he had an award named after himself in 1908. So his relations to the other scientists, the main connection between these three guys is that they work together at the Institute for Infectious Disease. These three men, along with many other researchers, work to create a basis for which we base our immunology studies off of. To be more direct, Koch and von Behring worked together on tuberculosis, and Koch, von Behring, and Ehrlich all worked on diphtheria. Koch's impact today cannot be overstated. He created a basis for studying bacteria. That's why he's called the father of bacteriology. His work led to the discovery of several harmful diseases and their causes, and not only that, the methods he used during this time were replicated, and we found the causes of typhus, tetanus, and the plague, among other diseases. Not only that, Koch's mix of staining bacteria and using an agar medium are still used in biology labs in order to look at the growth of bacteria.
Okay, my name is Devin Healy, and I'm going to be talking about Paul Ehrlich. And some facts about him is he was born on March 14th in 1854, and he was born in Strehlen, Germany. He died on August 20th in 1950. He was married in 1833 to Hedwig Pincus, and he had two daughters with his wife. For his education, he first went to the University of Breslau, Strasbourg, and also the University of Freiburg between 1872 and 1877 to study medicine at these universities. And then he earned his degree in medicine at the University of Leipzig in 1878. So he was a German-Jew medical scientist, and he worked in the fields of chemotherapy, hematology, and immunology. He received a Nobel Prize for Physiology and Medicine in 1908. So one of the things that he worked with was staining, and he was very interested in it. And so the problem or the focus for this topic is the use of staining methods on human blood cells and the use of dyes to stain cells. And so he knew that stain dyes had selective actions on various types of cells, and this suggested that chemical reactions formed the basis for cellular processes were taking place in the cells. And also Koch had discovered tuberculosis. And so the solutions and findings to these problems were that he developed different staining methods to differentiate the multiple types of blood cells in our bodies. He also found that chemical agents such as dyes could be used to repair dead cells and kill infectious agents. He also developed a new staining method after Koch had discovered tuberculosis. And so he developed a new staining method to identify tuberculosis, bacillus, and other mycobacteria. He was also focused in on immunology, and so the focus and problem for this was that he worked to standardize the process of the anti-diphtheria serum production, and this weakened bacteria was used in previous serum therapies like Pasteur's vaccines and Koch's tuberculin. And also, very little was known at the time about immunity, and so Ehrlich aimed to propose an explanation for the immunity process. And so, in doing this, he found or developed an effective immunization protocol. He created new serum therapies that use blood serum from immunized animals instead of the weakened bacteria that Pasteur and Koch had used. And he used these new blood serums to induce immunity. He also proposed a side chain theory for immunity, and this theory describes how antitoxins are formed and how they interact with toxins. So this theory states that cells present a set of side chains on their surface. And a side chain from a given cell may have a molecular structure that allowed it to bind with a specific toxin. And this binding causes the cell to lose its normal function, which in turn would trigger the production of additional side chains that would be released into the bloodstream. And these side chains would act as antibodies or antitoxins upon the binding to the toxin present in the blood. And this prevents the toxin from binding to other cells in the organism. And Paul Ehrlich was also very involved in chemotherapy. And so the focus for the studies was that he wanted to find an effective sera for serum therapy because it could not be discovered. He hypothesized that the relationship between the chemical makeup of drugs and their action in an organism needed to be studied. And related to this, he said that selective drugs were needed. And this means that he thought that we needed drugs that have a high affinity for pathogens without affecting host cells. And so his solutions to this were that he studied the treatment of trypanosomiasis and other protozoal diseases. And from this, he produced Trypan Red, which was effective against trypanosomes. After the cause for syphilis had been discovered, which was spirochete, he searched for a drug that would be effective against it. He created a new drug made from an organic arsenic compound. He named this drug salvarsan. He tested it on rabbits that had been infected with syphilis and found that it was effective. And so Ehrlich's work with Koch and von Behring, Koch had discovered the causative agent for tuberculosis, which was Tuberculosis bacillus. And Koch used, and so after this, Ehrlich went beyond Koch's studies and developed new staining methods to discover Tuberculosis bacillus. And Koch used weakened bacteria in the tuberculin that he created as a treatment for tuberculosis. After this failed, Ehrlich discovered the use of blood serum from animals as the new serum therapies. Von Behring and Kitazato discovered tetanus and diphtheria antitoxins. And from there, Koch invited von Behring and Ehrlich to work at the Institute for Infectious Diseases in Berlin. And so Ehrlich worked with von Behring to standardize the process of anti-diphtheria production. And so how Ehrlich's work and its impact on science and society today. So Ehrlich's staining method for Tuberculosis bacillus led to the development of Gram staining method for bacteria used today. Ehrlich's differentiation of the types of blood cells in the body using staining methods laid the foundation for hematology and the staining of tissues. And after Ehrlich's development of salvarsan as a treatment for syphilis, he became famous as the main founder for chemotherapy.